Good evening, doctor. I'm sorry, I have a question.
Good evening. Do you hear me?
Yes.
Чего у меня не записывается?
Doctor.
So this is microbiology lecture, microbiological control of pharmacies.
Doctor, I'm sorry, I have a question.
It's important to do microbiological control because of some medicines are prepared at pharmacies, and of course they should be sterile, and microbiologist control is carried out by bacteriological laboratories. Objects of study. Number one, distilled water for the preparation of medicines, except for drugs for injection and eye drops. Then distilled water for the preparation of injection solutions and eye drops, and injection solutions before sterilization. Eye drops prepared under aseptic conditions on sterile basis, which are prepared in the pharmacies themselves. Then object number three, the distilled water, 0.9% of sodium chloride solution. And then solutions of glucose, 5%, 10%, 25% and 40%, in which it is necessary to determine pyrogen forming microbes. Pyrogen is a substance, LPS, typically produced by bacteria, which produces fever when introduced or released into the blood. Pyrogens affect the central neurons, and sometimes it leads to shock. So we have to detect how many microbes, mainly gram-negative, are in these solutions because they are used for IV injections, intravenously injections. Number four, dry medicinal substances used for the preparation of injection solutions. Number five, pharmacy ware, stoppers, gaskets, other auxiliary materials, funnels, and cylinders. Number six, inventory, equipment, hands, and sanitary clothing. And number seven, air in the aseptic unit, in the sterilization, assistant, packing, material, washing rooms, in service hall. Object number one, distilled water for the preparation of medicines, except for drugs for injections and eye drops. Distilled water that is prepared at pharmacies. We have to detect the number of mesophilic, aerobic, and facultative anaerobic bacteria, abbreviation is MAFAN, and content of mold and yeast fungi, and coliform bacteria. Coliform, this term is produced from the species name Escherichia coli. So we are not looking for exactly Escherichia coli. We are not going to identify till the species name. We are looking for some bacteria, those belong to Enterobacteriaceae family and have the common properties with Escherichia coli. A detection of Escherichia coli indicates about fecal contamination. So just a few properties are necessary to study to make conclusion that coliform bacteria are detected. How it is possible to detect MAFAN and why this abbreviation is used? Mesophilic, aerobic, and facultative anaerobic microorganisms. Because of when we are going to detect them, the Petri dishes are incubated at temperature 37 degrees by Celsius. So we are going to detect just mesophilic microorganisms. Those prefer temperature from 20 till 40 degrees by Celsius. Some bacteria, psychrophilic, they need low temperature, so they're not able to grow up at temperature 30 AS7. Some microbes need high temperature, maybe 42, And so again, they are not able to grow up at this temperature. So we are not in past this we told about the total number of microorganism detection, but now this term is corrected. It's because of we are not possible to detect all microbes, just mesophilic microbes, and then the conditions for the cultivation of microbes in a Petri dish are aerobic, so the presence of atmosphere oxygen. So in such conditions, in aerobic conditions, grow aerobic microorganisms and facultative anaerobic. Strict anaerobic bacteria are not able to develop colonies in aerobic conditions. They are not able to multiply, they will die. So this abbreviation now is used: mesophilic, aerobic, and facultative anaerobic microorganisms. Not, of course, not total number of microbes. Anyway, just to make conclusion how many microbes are in this object, very often morphonym is used, detection of morphonym. How it is possible to detect? It is named sometimes plate count. This technique, this method is named plate count. We had to use 2 sterile empty Petri dishes, and the 1 milliliter of water is placed by 1 milliliter in one Petri dish and in another, and then melted and cooled till 45 degrees by Celsius. Mid-pepton agar is poured on it. Why the temperature exactly should be 45, maybe at least 49, because of 50 degrees, 55, 60 is too hot, and we will kill just microbes, those are in this water, and but not possible to wait the temperature below 45, because of a temperature 40, this agar again becomes solid. So, but we need melted agar because we have to pour it, and it is melted at temperature 90 degrees by Celsius, and we have to wait exactly till temperature 45. It's important. It is possible to pour agar, and we are not going to kill bacteria. So bacteria are still alive. Petri dishes are incubated in a thermostat during 24 hours at temperature 37 degrees by Celsius, and then 24 hours at room temperature. And then account the number of colonies grown on both plates, at first on the one plate, then at another plate, and calculate the arithmetic average. So finally, we have to, for statistics, we have to know arithmetic average. And so it will be the amount of morphonomy. The second method for identification mold and yeast fungi, the test water is inoculated in volume 0.5 milliliter on the surface of 2 Petri dishes with Sabourad glucose pepton agar and incubated at temperature 20-22 degrees by Celsius for 3-4 days. You know this medium, you did study microbiological cultural properties of fungi. In diagnosis of mycosis, very often Sabourad glucose pepton agar is used. And you know that fungi grow very slowly. It takes 3-4 days and maybe more. So we are waiting for colonies. And then we have to account the number of colonies, fungi colonies on 2 plates, and to find the arithmetic average. And then we have to estimate how many microbes in general, including bacteria and fungi. So the results are evaluated by the total number of microorganisms by summing the number of colonies of bacteria grown on the mid-pepton agar arithmetical average and the colonies of fungi. So we will know how many microbes are in this object, just in general. And then detection of the coliform bacteria. 2 methods are used. membrane filtration method, it is basic, and titration method. And we are looking for bacteria that grow on McConkey medium. You know, properties of Escherichia coli, they are cultivated on McConkey's medium to know lactose fermentation. And we have to see lactose fermentation, colonies with the color of indicator. And then they are by microscopy, they are gram-negative rods singly arranged, and there are oxidase negative bacteria, and they are able to ferment glucose also. But if bacteria with such properties are detected, we can make conclusion that coliform bacteria are detected. So no antigenic properties in sanitary microbiology, the methods are very simple. So to know morphology, staining properties, lactose positive, glucose positive, and oxidase minus, that's enough to make conclusion that they are coliform bacteria. Then standard, then we should compare results with the standard. Standard is the maximum allowable content, 10-15 microbes in 1 milliliter. Content of coliform bacteria in distilled water is not allowed. Sometimes it is necessary to make microbiological control of purified distilled water. In this case, we are going to detect the total number of bacteria, again morphonema, and bacteria of Enterobacteriaceae family, and we are looking for Pseudomonas aeruginosa and Staphylococcus aureus. So in purified distilled water, by standard, it is acceptable to have no more than 100 microbes in 1 milliliter, and no bacteria of the family Enterobacteriaceae, Pseudomonas aeruginosa, and Staphylococcus aureus is not allowed. Then the second object, distilled water for the preparation of injection solutions and eye drops, injection solutions before sterilization, and then eye drops prepared under aseptic conditions on sterile basis, which are prepared in the pharmacies by themselves. In this object, it is necessary to detect the number of mesophilic, aerobic, and facultative anaerobic bacteria of fungi. And you know this method, I did explain you. And then the number of bacteria of the group of Escherichia coli, again, McConkey's medium, inoculation of McConkey's medium. Then we are looking for lactose positive bacteria. Then we have to do microscopy, and they are Escherichia group of Escherichia coli bacteria, gram-negative rods, and they are oxidase minus, and they are able to ferment glucose also. So, and by standard, the number of fungi we should detect according to the special table with standard, and the presence of bacteria of the group of Escherichia coli not allowed. The third object, distilled water and solutions for IV injections. Like sodium chloride solution 0.9 and glucose solution, different concentrations, 5, 10, 25 or 40%, in which it is necessary to determine pyrogen forming microorganisms. Because of these solutions are used for IV injections, and 3 methods are used to determine pyrogenicity: biological by using of laboratory animals, indirect, and lab test by using of some fishes. So a few methods are detected. And what does it mean, pyrogenicity? We are talking about gram-negative bacteria. They have LPS in the cell wall that is able to cause fever and high temperature when it is circulated in the blood. Such ability is named pyrogenicity. And unfortunately, LPS is heat stable, is not destroyed by sterilization. Of course, these solutions, they should be sterilized for sure for IV injections, but it is possible to kill bacteria, it is possible to destroy bacteria, but not possible to destroy lipopolysaccharide. Anyway, it will be injected with these solutions, and there are different clinical forms, and maybe caused by LPS action, maybe mild form, maybe very severe form. Sometimes it led to shock and even to the lethal outcome. So we have to detect them, how many microbes are in these solutions before sterilization. Then number 4, we should exempt dry medicinal substances used for the preparation of injection solutions. Sampling of dry medicinal substances is carried out with sterile spoons in sterile laboratory dishes in the amount of 30-50 grams. If the substance is tableted, then it is taken with sterile tweezers in a sterile laboratory glassware in the amount of 30-50 grams. And in this case, it is necessary to detect the number of morphonomy and the presence of bacteria of the group of Escherichia coli. You know morphonomy detection, again, by using of 2 Petri dishes. We should place 1 milliliter of this substance, 1 gram, and should be in the water. And then we should pour by melted agar, and then we have to count number of colonies. Quantitative determination of bacteria of the group of Escherichia coli. 1 gram of drugs is inoculated on a dish and poured with endomedium, maybe McConkie medium, the same diagnostic medium that content of agar, lactose and indicator plus alkaline, and using the deep inoculation method. Inoculations are incubated at 37 degrees by Celsius for 18-24 hours, after which colonies typical for bacteria of the Escherichia coli group are taken into account. Typical colonies are, of course, lactose positive colonies. They have color of indicator due to lactose fermentation. Standard, the number of morphon should be compared with the special table. The presence of bacteria of the group of Escherichia coli not allowed. Then 5 pharmacy wear, stoppers, jackets, other auxiliary materials like funnels and cylinders that are used in pharmacies. The study of these objects is carried out by rinsing with sterile tap water in a volume of 10 milliliters. In the flushing fluid are determined again the number of morphonomy and the presence of bacteria of the group of Escherichia coli. Standard, the amount of morphonomy should not exceed 150 colonies with 3 bottles, 5 stoppers, and 5 gaskets, means 10 milliliters of flushing liquid. The bacteria of the group of Escherichia coli not allowed. Number 6, inventory, equipment, hands and sanitary clothing. Study the list of objects subject to control by washout method. Table for preparation of injection solutions, eye drops preparation table, scales for weighing dry substances at the defector, container for storing gaskets and stoppers used for filling injection solutions and eye drops. So different objects should be exempt in pharmacies that are used for preparing, and hands should be exempt. Washings are carried out using cotton swabs placed in test tubes with solutions of sodium chloride or 1% pepton water. In the flushing fluid are determined bacteria of the group of Escherichia coli, then pathogenic Staphylococcus by indications. Standard, Escherichia coli bacteria and pathogenic Staphylococcus bacteria are not allowed in washes. Then the 7th object, air in the aseptic unit, in the sterilization, assistant packing material, washing rooms, in the service hall. The air at different rooms in the pharmacies should be exempt for microbes presence. Air sampling is performed under the following conditions: clean room prepared for work, closed vents and doors, determination of the percent of relative humidity in the room, air sampling height level corresponds to the height of the desktop, not earlier than 30 minutes after the wet cleaning the room. We have to do air examination. Air samples are taken by aspiration method using devices for bacteriological air analysis. There is special equipment for sampling of the air. It works like vacuum cleaner. So it takes the air, and it is possible to know how many liters of air are exempt. Do you see it as a top? And it should be opened, and we have to place Petri dish with medium inside, and it has a hole in this top, and the Petri dish is rotated. The air goes through this hole, and particles of dust with bacteria are inoculated on the medium in Petri dish. We are going to detect total bacteria content and the quantitative content of Staphylococcus aureus and the quantitative content of mold and yeast fungi. It is possible to set the speed of inoculation of air blowing speed 25 liters per minute for specific analysis. And then we have to how many the amount of air is determined. 100 liters for determining the total number of bacteria, 250 liters for determination of Staphylococcus aureus, and 250 liters for the determination of molds and yeasts. Petri dishes with inoculations on for detection of total number of bacteria, just nutrient agar is used, ordinary basic medium. For determination of Staphylococcus aureus, yolk salt agar is used. You know, it is selective medium for Staphylococci isolation. And for detection of molds and its Sabourad medium is used after inoculation. So Petri dishes with inoculations on nutrient agar and yolk salt agar are incubated at 37 degrees by Celsius for 24 hours. Inoculations on Sabourad medium are incubated at 22-28 degrees by Celsius room temperature for 4 days. For determining total bacteriological contamination, the colonies are examined after 48 hours, and the number of grown colonies is counted, and the count is made per 1 cubic meter of the air. A quantitative content of Staphylococcus aureus, the colonies are viewed on yolk salt agar after 48 hours of incubation. Suspicious colonies are identified. So suspicious colonies of Staphylococci are S-type colonies with pigments. They always produce own pigments. We have to account colonies, and again the results obtained are recalculated per 1 cubic meter of the air. The amount of mold and yeast fungi, the colonies are incubated for 96 hours, then the number of grown colonies is counted and then recalculated again per 1 cubic meter. Standard, the total number of colonies of microbes in 100 liter is allowed in all rooms. Then Staphylococcus aureus should not be in 250 liters of air in all rooms, except for the service hall. In service halls, of course, there are many people, and again, it is just up to 100 for 1 cubic meter. Molds and yeast fungi should not be in 250 liters in all rooms, except for washing, up to 12 in 1 cubic meter, and service hall up to 20 in 1 cubic meter. So the lecture is over, and I have some questions for the leaders. Where I will send you examination questions and examination procedure. If you will get some question, I will answer your question. And is it possible to join? Next week you have no lecture. Have you any lecture on 25th of December? Is anyone able to answer me? Ashraf,
good
are
evening.
you able?
Yeah, can you hear me?
No, on 24th. I'm sorry. Now it is 17th of December.
Yeah, no, we don't have any lecture. No, we finished. We all
Ashraf,
finished.
do you hear me? But
Yes,
I didn't
yes.
hear you. Could you write me тогда? Okay, so нет, надо. I don't hear
Yes.
you. Вот.
Here, can you hear us?
Yeah.
And me now?
Don't have.
Yeah, here too. Yeah.
Okay, okay. We don't have any lecture.
So I will send all leaders examination questions and examination procedure. Then you will think about it, and we will meet on 24th of December at the same time. Is it okay?
Yeah, it's okay.
4:50. Of course, I should answer your questions on January before exams, but on January we have holidays till 10th of January, and then from 10th of January you have to take exams. So no working days on January, and so we have to meet on December. Okay.
Okay, we will discuss just the form of the exam. Just the form, like.
Sorry.
We will discuss just the form of the exam.
The procedure. The procedure.
Okay.
The form of exam I will explain you. And if you have some questions on microbiology, on the questions, I will send you questions, examination questions. But really, there are no new questions. They are the same questions that you did get for test number 1, 2, 3, and so on. They are the same questions.
Okay, it's MCQ.
It will be MCQ, but it will be, I hope, it depends on epidemiological situation. Of course, you know, again, new variant was developed, Omicron, and but yes. I hope that we will meet anyway at medical university, and you will take them at lectures hall, 2nd floor, and it will be MCQ, and you did answer questions. Do you remember you did take tests for the lecture? So you know approximately what are these tests, and then you will get a question about some preparations, some medicines. Okay.
Okay.
I will send you a list of these preparations. Did Pavel sir send you, inform you?
He sent me something. He sent me preparations. It's a list of 12 questions. Vaccine, serum, immunoglobulins.
Yes, yes, just in general.
Yeah, but not in details.
Really, нет, there are, you should realize in general vaccines, and on them you will get any vaccine. So, and you should describe what it is prepared of and practical application. So you should know all vaccines, repeat, of course, what types of vaccine, and you did study, really you did study vaccines. Then serum, immunoglobulins, you should realize what are they. And we have a lot of immunoglobulins. You will get this certain name, like for example, Staphylococcal immunoglobulin. And you should know that every immunoglobulin contains antibodies. It is a purified and concentrated preparation that is prepared of hyperimmune serum of blood, maybe either donors, human beings, or maybe animals, maybe homological immunoglobulin, maybe heterological. Unfortunately, horses are used sometimes for immunization because we have no donors enough. And you have to know what is it. And if we will get staphylococcal immunoglobulin, should realize there are antibodies. And in case of staphylococcal infection, there are antibodies for exotoxins, okay? If it is immunoglobulin against tick-borne encephalitis, this medicine contains antibodies for the virus, exactly, positive agent of tick-borne encephalitis. So you should know in general diagnosticum, what is the diagnosticum, and bacteriophages. You will get any bacteriophage, maybe typhoid fever bacteriophage, maybe dysentery bacteriophage. So you should know in general what are they. Bacteriophages, they are viruses or bacteria. And then in accordance with the name, you should realize practical application. So you should know in general. Do you understand me?
Yeah, so we will,
So
we
I'm
will
not
have,
in medicine. A lot of preparations are used. I'm not able to send you all of them, and they are written in textbook. Okay, the information that you did get for test number 4, 5, 6, 7. Okay.
okay, so we will receive real medicines, and we will comment. We will say what's there, and
I will explain you. Okay, we will meet.
Okay.
Okay, next week we will meet at 4:50, and I will show you the list of these preparations and what should you write. Okay.
Okay, thank you.
What answer should you give? So it is. It was the last lecture, and I hope you did get knowledge. And if student didn't pass the previous test, it is possible to do on January, exactly on the day of exam. Every student has just one attempt on January, and then maybe later on March, I don't know. If student is ill, is not feeling well, student should go to the doctor and get the excuse that really gets proper that the student was ill, and Dean will give them additional time on March. Okay.
Okay.
If you have no questions, I will finish the lecture. So
Okay,
memorize,
so
we will
much.
meet, inform all your group mates. We will meet next week.
See you next Friday.
We will meet. Bye-bye.
Bye-bye. Have a nice evening.
Bye, you too.